you're fast. Look at you. All right. So onwards and upwards to a new unit. This one should also be familiar. So we just sort of wrapped up our genetics in lab. A lot of what we do here is going to feel really familiar. Keep in mind as we do this, right, this is going to be referencing both our labs that we had. So we had our first classroom lab, right, with the videos, which would be also very helpful review videos. We took notes on the allelic relationship and did some practice Punnett squares. And then we had our dragon genetics lab, where you flip the sticks things. I was going to call them toothpicks, and that definitely wasn't it. And then had a few practice Punnett squares, but mostly things like definitions and thought problems. So all of that stuff is going to help you here. Also keep in mind that those things, if you have not done them already, will come due here. The dragon one will probably, if not already, be due. And the genetics practice problems come due as soon as we're done with this. We'll be doing some more practice problems to help reinforce that. So if you have not done that stuff already, do keep an eye on the deadlines there so you don't just forget they ever existed. Okay, so like before, we're always going to start with a little vocab, right, which again will probably feel very familiar since we've been using this stuff for the last couple weeks. Then we're going to look at the principle of segregation, which is something that you have been doing for a while now in meiosis. So again, something we're just going to put a term onto. The next couple of bullets then, what I'm going to do is we're going to work through setting up the foolproof Punnett square, which you've had a little bit of practice with on your own, but I want to go through together to make sure that we have all the checkpoints down. And then what we're going to go through is work through a big working in-class activity together to practice a bunch of the different allelic relationships together. So you did, again, a little on your own. We're going to do a lot together. And then depending on time, we're going to work through some family trees and look at that genealogy, allelic relationships, and then some more definitions that work with meiosis. Okay, so let's look at some terminology. I'm going to flip this. I'm going to pretend to flip this to the other side. So we've been using the word gene for quite some time. But at this stage, we want to make sure that we're using it absolutely correctly and with confidence. So remember, as we're looking at this stick set here, that this double stick set is a chromosome, right? A duplicated chromosome. A chromosome is a subunit of DNA. DNA then being all of the information that you need in the cell, right? That's our information, the books inside our library. So when we're talking about a gene, okay, we're talking about information, right, within one of these books, right? A code for something. Okay. So when I say a code for something, okay, an example of that might be eye color. Okay, so here's the gene for eye color, or the information on how to make eye color. Perhaps this one is hair color. The gene on how to make hair color. A gene on how to make height. Well, that's not even remotely how you spell that. Now, we've talked about this before, right? There are genes for just about everything. Okay, genes on how to make your heart pump, genes on how to make bile in the liver, right? So there are lots of different types of genes, and these all code for something. Okay, so we're mostly going to talk about simple genes like this that make obvious or easy things. But keep in mind that there are lots of different types of genes. All right. So if this is my gene, okay, this is my code, my section of information on how to make eye color, then an allele are different versions of that gene. Okay, so we think about there is information on how to make eye color, okay, but there's different eye colors, right? I can have blue eyes, I can have green eyes, I can have brown eyes, I can have hazel eyes. So there's different versions or variations on that gene. And so, look at the example here. The other thing you need to keep in mind, and that's going to become very important, is this is a chromosome. And our chromosomes are made up of sister chromatids. So you have two sister chromatids for every chromosome, which means you have two copies of every gene, at least. Which means you can have up to 2 alleles for every gene. So if we focus on the top one that we've been working with, okay, this is my gene for eye color, and I happen to have 2 different alleles for eye color, right? Let's say this is the eye color or the allele for blue eyes, okay, the variation of the eye color gene that makes blue eyes. Okay. And let's say this is the allele that makes green eyes, the variation of the eye color gene that makes green eyes. Okay, they're both eye color genes. They're both the same gene, but one makes blue eyes and one makes green eyes. With me so far? So let's look at these alleles a little more. So if I have 2 alleles, 2 variations of a gene that are different, like my eye color one, you see I have a blue one and a green eye color one. So those are 2 different alleles. Then I have heterozygous gene. So hetero being the prefix for different. So heterozygous alleles. They're different. I have one blue one and one green one. If I have allelic copies that are the same, so if we look at my hair color gene, okay, this allele makes blonde hair, and this allele also makes blonde hair. Okay. If those 2 copies are the same allele, then I'm homozygous. Homo being the prefix for same. Okay, that's not so bad. These terms are kind of familiar and for once a little intuitive, right? Better than chromo, chromo, chroma, right? Right. So now let's look at the relationship between our alleles. Okay. So very generally, alleles can be described as dominant or recessive. So if we're looking at a dominant allele, this allele is normally described by a capital letter because its expression is big, bold, and loud. Okay. So you've heard me talk before about how this is the really loud guy or gal at the party. Okay. So no matter where you are, okay, you can hear them. Okay. You're standing at the chip bowl, talking very loud. You only need one of them. And no matter where you are in the room, somehow you seem to be picking up snippets of their very obnoxious story. So this is what we mean by a dominant allele. Right? They have such a loud personality, okay, that it dominates the space. Okay, so no matter what else is going on, you're getting pieces of that. So if we look at a situation like this, where we have only one dominant, we see that we're getting the dominant expression because it's so loud. Okay? It tends to cover up what everybody else is doing. And that's what dominants do best, right? They cover up everything else. When you see a dominant, you know it's covering up, it's masking everybody else. Now, comparatively, our recessives are shown with lowercase or little letters. They're quiet. They're small. Now, be careful. This doesn't mean that they're less important, okay, or less frequent. It just means that they're more easily covered up. Okay, so there's a really big difference there, and we want to be careful about that. Okay, so our recessive person, right, is the person at the party that likes to sit in the corner and talk to the cats. Right? Okay. Now that person still might be super interesting, okay, but there's only one way to talk to that person, right? And that's if you also, so if a second person sits down with them and talks. So you have to have two people quietly sitting down in the corner to hear them or have a conversation. And that's the key with the recessive allele, is you need two of them for them to be heard. Right? Because they're easily drowned out, right? They're easily covered up by whatever other activities are going on. So in this case, right, we needed two of them to be able to see that expression. Otherwise, they just get covered up by the loud guy or gal in the middle of the room. Dominant versus recessive, very clear? We're going to use those a lot. We're going to twist them around a lot. So. All right, one more. As we think about these final allelic relationships, the information they give us is really important, and they give us a lot of information, right? We have a lot more now than we had at any previous stage. So now we have what's called the genotype and phenotype of an organism. So the genotype is the actual code of those alleles, right? So I know I have, say, one dominant and one recessive. So the actual codes for that. I have a capital and a lowercase, the representation, those codings. Okay, so I want to say genotype, right? The codes of the alleles of that gene. This will be whatever letter system that you're using. You're actually going to write that code out. That's your genotype. A lot of very specific information there. So this person is big B, little b, right? Dominant and recessive. I am heterozygous. Okay? All of that stuff is my genotype. My phenotype is the translation of that information, right? The physical expression of that code. Okay, so if I use my key down below, I have one dominant, one recessive. I know that dominant is a bully. We're going to cover up. I talk really loud. That means I'm going to have brown eyes. And that physical expression, the having of brown eyes, is my phenotype. So phenotype, physical. Okay, so the genotype will tell us what our phenotype is, right? It's like decoding a message. So good? Okay, so let's go through one more definition that we need. The principle of segregation. Again, this is something you know. We haven't actually put words to it in the past. So when we were doing meiosis, I remember that we ended with 4 haploid daughter cells. Right? And each daughter cell had 1 allele in each, right? AKA 1 sister chromatid in each. And that's the way we always drew it, is it got 1 sister chromatid. Okay, but now we know, as we think about each sister chromatid has 1 copy of each gene, right? And therefore, getting 1 copy of each gene, aka 1 allele in each box. So that's all we're saying now. Instead of coloring these things in, we're saying, well, we know those colors represent alleles. Right? So instead of mom's copy just being the red chromosome, and we know that that is either a big A or a little a. Okay, so we understand that that's representing a version of a gene. That's going to make me tall or short. That's the information on the red version versus the pink version of the chromosome. So there's key information that we've been doing all along we haven't been paying attention to. And notably, our alleles aka our genes, aka our sister chromatids, are discrete units. Right? In other words, not confetti. So we're not taking those genes or alleles, ripping them up and just sprinkling them in our daughter cells. Right? We moved those sister chromatids as whole pieces. Our genes moved as whole units, which is good, right? If you tear a gene in half, that's not bad because you can't do it anymore. You lose that information. So by discrete units, we mean we're moving them as whole pieces. We're not tearing anything up and sprinkling it away like confetti, right? Little sheets of paper. And we also knew this, that you're starting with 2 alleles for a gene, which is the same thing as having 2 chromatids, which is the way that we drew it. And now we just have to recognize that's what it is. Okay, and that the whole point of meiosis, both in metaphase 1, when we separate mom and dad, right, into 2 separate daughter cells, and in meiosis 2, right, when we're separating the sister chromatids into separate daughter cells. The whole point is just to divide these up randomly and equally into these haploid gametes. So this isn't ultimately anything new, right? We just have to think about, well, what have we done all along? Okay, so we weren't shredding up chromosomes all along, right? We knew that we had 2 copies of chromosomes of each, right? That's what being diploid means. Okay, so we had all these pieces, right? We're just adding a definition now. Any questions so far? All right, so I'm just going to introduce the Punnett square, and then we'll actually start to use it next time. Okay. So the key with the Punnett square, remember, my goal is what we're going to do is set up the foolproof way to do it. And the key with the foolproof way is it sets up several checkpoints for you. You can always go back and say, is this right? Okay. Or have I fallen into a pitfall that you can use at any stage? And then we're going to use it a lot. So the key with the Punnett square is, as we look at the general setup, is I'm going to take mom, I'm going to break down her possible alleles, all of her possible allele combinations, and those are going to go across the top, always. Now we're going to keep track. Ladies across the top. I'm going to do the same thing for dad. I'm going to take his possible allele combinations. I'm going to always put them down one side. Okay. Ultimately, these make all possible gametes. Right? So technically what we're going to set up is all of the possible gametes that would come out of telophase 2. In the middle, then, what's going to end up is the theoretical offspring from fertilization events. Now, it's key to remember that these are possible, not promised children, right? So we're making all of the potential babies that mom and dad can have, not that they will, in fact, have all 4 or all 8 or all 32 of these children. Right? So this is not guaranteed, but one of the children, if they have them, will have to come out of this set. Right? So potential bebés are what we're making. Okay, we're going to set up an example and work through it together very slowly next time. But certainly we're going to stop there today.